Neuronal networks are spontaneously active, even in vitro in brain slices. When researchers record spontaneous glutamatergic currents from a single neuron, it gives them information on the activity of the glutamatergic neurons afferent to the one being recorded.
I record AMPA currents in the whole cell configuration and in voltage clamp mode. I add gabazine to the extracellular environment to block GABA-A currents as well as APV to block NMDA-type glutamate currents. In this experiment, the cation reversal potential is positive 10 millivolts. If I hold the membrane potential at negative 70 millivolts, AMPA current will be negative. We see here currents that show a downward deflection of the baseline. These currents are negative and therefore inward. A time base of about 40 milliseconds gives us an indication of AMPA current's duration and frequency of occurrence. We see a recording of spontaneous AMPA currents over time passing here on the screen. These currents have a variable amplitude on the order of about 10 picoamps because we do not always have the same number of active glutamatergic synapses at the same time. Thus, in effect, AMPA synaptic currents result from a simultaneous activation of multiple postsynaptic AMPA channel receptors by a glutamate release from the axonal terminals afferent to the neurons being recorded. To make sure that we are, in fact, dealing with an AMPA current, we add CNQX to the extracellular environment, which is a specific antagonist for the AMPA-type channel receptors. We then observe a gradual disappearance of AMPA currents. We launch automatic current analysis in the software and then validate this analysis manually. The parameters of interest to us for each current are the magnitude of the deflection here between the yellow point and the red cross, the time between the yellow point and the same red cross, and then duration between the red cross and the violet point. We are also interested in the AMPA current's recurrence frequency, which may be measured by the time between two yellow points. The diagram on the left shows us the AMPA current amplitude. Here, for each blue point, we have the measure of the mean AMPA current for a given cell, or for an experiment. The red line indicates the mean amplitude of all the currents, about negative 18 picoamps in this case, while the two green lines represent the variability, i.e. the standard deviation. The diagram on the right represents the inter-event interval, IEI, that is to say, mean time elapsing between the appearance of two consecutive AMPA currents. Each point represents the mean IEI for a given cell, and all the points represent the mean values that we recorded during our experiments. The red line is still representative of the mean, about 4 seconds, while the green lines inform us of the variability. This helps us understand the activity of AMPA-type glutamate synapses afferent to the neuron being recorded. This activity may vary with development or pathology.